And I'm like, okay, well, hydrogen.
Yeah, I don't. So I don't want to.
Every one of them has kids. This way it used to be. Not to mention so he's just now kind of last time I talked to him he was kind of stressed out about everything, kind of decided exactly what he was proposing.
Actually, you don't feel okay.
Hello, good morning everybody. So we're gonna start in 1 minute, 30 seconds. Now. So it's a great pleasure. I want to introduce you to Dr. Sagar from University of Florida. My connection with him is we share interest. He is a medical doctor, I'm a biologist, you're all engineering students, but we all share the passion of biological and medical issue, the problem that can be solved preparing machinery to study whatever biological system you're studying. So he came to the United States in 2000 after he did a degree in medical and gastroenterology in India. Then he came 4 years for a fellowship at Yale, and then another 5 years Rochester University, and then in 2010 he moved to University of Florida and is a senior professor right now. And today he's gonna show you some fun stuff. I was able to participate 5 years ago with machinery to study and to practice whatever you want to discover with the drug, some case, or with the special formulation that he created with this new technology to use amino acid formulation to induce or repair a tissue like endothelial tissue, guts, the skin of the lung. So it's my pleasure to introduce you right now. We're going to start the talk and everybody can see. Thank you.
Can you hear me? Did you put it on?
I didn't turn it on.
Oh, you didn't turn it on?
No, I didn't turn it on.
Hello, can you hear me? Yeah, good.
Yeah, here and here, not there.
Good morning. You know, First is thank you for inviting me here, and do I need a mic?
It's for this room, I think.
Let's see, I just keep it.
But it's for the Zoom, because they won't be able to hear you on Zoom if you don't.
If I don't use the mic? Okay,
So
so,
this room doesn't have real good acoustics.
okay, all right. So, as Daniel just mentioned, my background is medicine, but I do have a PhD in electrophysiology, and I have a deep passion for math and physics. So just saying it, but I'm not formally trained in math and physics. I'm going to learn from you guys here. That's one of the reasons I'm here. So let's start the talk, but first of all, I should thank the biomedical engineering for inviting me here, because I think this will open up some channels for new discoveries. So saying that, the title of today's talk is Beyond Bench to Bedside. So the work that we do in the bench, how do we take it to the patients? And actually there is one more thing that should be there, that is bench to bedside and back. So we make things possible for patients and we learn from patients so that we improve the machine. So that is bench to bedside and back. And the most important thing is that, again, from my personal experience, is that you need biomedical tools. And there is a crosstalk between the doctors and the engineers and the scientists to decode certain disease conditions. So this is how I will open my talk. So this is my U of team. This is University of Florida, Gainesville. I should also thank the funding sources. We have funding from NIH. We also have funding from some of the companies, that is the spin-off companies that we have created. Intrinsic Bioscience is a company that we founded way back in 2010. 352 Ingredients is a company we launched at the beginning of this year. We already have licensed quite a formulas from this. And one of the things I will say here is that as we go through this talk, you can stop me at any stage. Something is not clear. I want to keep this as engaging as possible because we come from two different backgrounds. So please ask questions. So I do have conflict of interest. I'm a shareholder in Intrinsic Bioscience, in 352 Ingredients. And also we have a license over 170 plus patents into various companies, and that's all from University of Florida. So University of Florida Research Foundation owns those patents. We have licensing terms with a lot of companies, starting from Nestle, Johnson & Johnson, Reckitt and Benckiser. So these are some of the deals that we have signed. I will walk you through this, and this might entice some, what is it called, thoughts in the work that you actually do. So before we go into the presentation and understanding the bench to bedside concept, I think we need to understand why do we need to study this. So we need to identify the gap, and how do we study the gap, and that is what I'm going to focus on today. So for me, I'm looking at the gaps in clinical practice. I'm sure in biomedical field, you may have gaps in the tools that you all use. Those are all the areas that we need to improvise. And that's what we're going to, and that's taking you to the problems and solutions. So identifying gaps. So as I said, I'm a physiologist by training now. So every non-disease conditions, there may be physiological alterations. Let's say you have a diarrheal disease. Why do diarrheas happen? You have electrolyte loss, there is active, there's a channel called chloride channel that secretes chloride. Once salt is lost, water will follow salt by osmosis. So you have salt and water loss. You have sweat, again, you have water and salt loss. So now, what is a physiological phenomenon that is happening there? That is a chloride channel. So if you can regulate the chloride channel, you can prevent diarrheal diseases. Now, in certain situations, it is secretion. In certain other situations, it may be an absorption defect. Or it may be a combination of secretion and absorption defect. Now, like cholera, you have secretion defect and you have an absorption defect. You lose around, say, 15 to 16 liters of fluid in 24 hours. Your patient can die right in front of you in 6 to 7 hours if you don't treat them with sufficient fluids. So that is a physiological function we are talking about. Now, we talk about biochemical assays or discovering a key biomarker. So we need a marker. So like I said, chloride channel. It could be a tool, it could be a a biological protein, and that could be used as a marker to regulate the physiological functions. Now the question is, okay, you have identified a protein, or you have identified a channel. How do you regulate them? Do you have necessary tools to study that? As a physiologist, I can tell you, in 60 to 70% of the situations, you don't have a tool to study the physiological function. We have to make it. So Damien here, and we here also host to all this. Any physiologist in this room will actually talk about it. We don't have ready-made tools. You have to make the tools, and that is where engineering will come into focus. Now, once you make the tools, I'm not an engineer. I had to get engineers who are formally trained in the business and real-time data acquisition. So once you make the tool, some data is coming out. It could be junk, it could be meaningful. How do you find that is meaningful? So analysis, interpretation, again, you bring smart people into it and you start making tools. And I'll tell you that we have aerospace engineers working in my department, and you will be thinking, what is an aerospace engineer doing in medical field? But they do. And once you do all this, you have to make sure that this is still working in a whole body system. So you have to have an animal model. So and selecting the animal model is another critical step before you can translate this to a patient or human application. So unmet need. I'm going to go through 3 of them here if I have sufficient time, or I'll just go with just 2. These are things that we have developed in the lab, we have commercialized it, and we of course get some royalties because of that. University of Florida may be gaining out of that too. So the first formula was the entry that was made for overcoming the gastrointestinal complications in patients who are receiving radiation chemotherapy. You know, when patients get radiation or chemotherapy, they have nausea, they have vomiting, and they have diarrhea. So how do we prevent that, or how do we minimize that? So that's the first formula we developed. My passion has always been diarrheal diseases. We have worked extensively in various countries, and we developed an amino acid-based oral rehydration solution. We have done clinical trials with the help of WHO, that is the World Health Organization. We'll talk about it. Peristalsis, we will talk about peristalsis, another machine that we very recently developed, licensed out. We made a formulation on it. It's on clinical trials now. Then we also have lung mimetic systems, you know, cystic fibrosis, allergic rhinitis, asthma, you know, all these kind of conditions. How do we make tools? So we're going to talk about that a little bit. So as I said before, identifying the biomarker is an important step, and you need, I would say you need innovative concepts. And we've talked about real-time data capture and how do you develop assay systems. So having a biomedical tool, having a bioassay system, and how do they both integrate into one system. I think you need both a biologist, a doctor, as well as an engineer coming into this. So I want to start the story by some work that took place even before I was born. Let's put it like that. So way back in 1960, a simple biomarker or a protein was discovered that is called SGLT1. All it does is it brings 2 molecules of sodium for every glucose molecule. One sodium is absorbed, water will go into the body by osmosis. So you're absorbing sodium, salt, and you're bringing in water into the body. And that was a phenomenal discovery that went ahead to save 5 million lives even today. So children don't die because of diarrheal diseases, because of this major discovery. In 1966, a formulation was put together, and by 1968, it was tested in Bangladesh. And by 1978, WHO adopted it, and we saved millions of lives today. A simple discovery, but you need a tool to study this, and that's what we're going to go into. But even with this saving of 5 million lives, it is heavily underutilized. Less than 3% of the population with diarrheal diseases actually use this formulation for various reasons. Number one, it does not reduce stool frequency or stool output. That means a child who is having diarrhea and you give oral rehydration solution, you suddenly see an increased stool volume coming out. And moms don't like to see their children having an increased poo after giving a treatment. As simple as that. So if you just look, it is heavily underutilized. Less than 33% of the population with diarrheal diseases use an oral rehydration solution. That is a gap. How do we confirm this is true? People talk about it, but how do you validate that? So we took a team. And we went straight to Bangladesh, where this formula was first developed, and we did clinical trials with the help of WHO in a center called ICDDR, the International Center for Enteric Diseases. And that is a WHO referral hospital where severe children with severe diarrhea come in. And what did we find? We measured the stool volume, and we found that you use oral rehydration solution that contains glucose, it saves life, but stool output is more. And it does not prevent future episodes of diarrhea. Just because you took a treatment today, it doesn't mean that tomorrow if you get a diarrhea, the stool volume is going to be less. So these are where the limitations. So in our lab, what did we do? We wanted to study why did glucose fail to reduce stool volume. And we found that glucose, in addition to that, I may be using this a little bit so that I can, so glucose, in addition to the chloride secretion, it is also stimulated, in addition to the sodium absorption. I'm going to draw a picture here. Let me see if it will over this. Never use this. So, you know, there is something like this. Have you seen this? This is a villi in the gut. There are finger-like projections out there with cells lining on this. So this is a villi, and this is a crypt. Crypt is secretory. Villus is absorption. All the sodium, all the salt absorption happens in the very top. And in the crypt, they have immature cells, they are secretory in nature. The glucose absorption, the sodium absorption happens in the villi, whereas secretion happens in the crypt. What we found is glucose stimulates chloride secretion. So anybody coming with diarrheal diseases, the net fluid is the absorption from the top and the secretion coming from the bottom. So if the secretion predominates, then the net fluid that is getting into the body is actually less. So now there are disease conditions where villi may be smaller, like in radiation, chemotherapy, tropical sprue, Crohn's disease. The villi are blunted, they are shorter, but the secretion from the crypt will predominate. So in these situations, a glucose-based oral rehydration solution cannot be used, or it has got minimal effect. And that gave us a gap. And that gap, we made a formulation. So you can see here, we have the villi, we have a crypt, we have the stem cells, they divide, they produce daughter cells, they migrate up. And this is the publication that we showed that glucose increases intracellular calcium and produces a chloride secretion. Then, for the oral rehydration solution, we studied how does, as I said, the villi is blunted, the crypt is still secretory in nature. So in patients who are receiving radiation or chemotherapy, the villi is much, much shorter. Secretions will continue to happen from the crypt, and therefore we needed to have a solution that will enhance absorption even when the villi is shorter. And that is what we did. So what we did, we looked at disaccharides, we looked at proteins, we looked at dipeptides, we looked at amino acids. We found there are 7 amino acids that will bring sodium into the body, but some of those amino acids will also stimulate secretion from the crypt. The end result is that we have a 5 amino acid formulation that will bring sodium into the body, will not secrete chloride, and also tighten the mucosal barrier. That was the first formulation we developed, and we went ahead and did clinical trials. So, with the help, this was a study that we got a major NASA grant, and this is an electron microscope picture. This is a normal tight junction and an adherence junction, and you can see that when you give radiation, even the adherent junction opens up. Now, if you give the right amino acid formulation with the right amino acid that tightens the mucosal barrier, you are able to tighten this. So this is all working in animal models. But can this be translated into a patient? So we put this in a border and we called it N-Trade, and we started doing clinical trials. And before we do clinical trials, we wanted to have an animal model in which we wanted to see, can you give like frying doses of radiation and see if the gut is damaged, and can you reverse it? And that was, and can the animals survive after the radiation? And that was a study that we did on this animal models. So we gave radiation doses like 1 gray, 3 gray, 5 gray, 7 gray, 9 gray. And you know, in a normal radiation that you give to patients is around 2.5 to maybe 3 max, but we went up to 9 gray to give sufficient doses to destroy all the cells in the body and see if the animals can still survive. So we did, once we knew that the animals were surviving, we did allometric scaling. We can talk about that in detail, but I'm not going to go about that here. So we tested this in patients, and we succeeded. And these are massive clinical trials that we did in various places. So our team also felt that this could also be used in sports. So we also made in acute hypertonic and isotonic dehydration. So the story is very simple. We went to the army. We got this army subjects in a heated chamber, and they were running on a treadmill, and they were made to exercise to lose body weight up to 3 kilograms of their body weight, and they were rehydrated with different fluids, and we proved that we could rehydrate, and we looked at all the plasma markers to see what is the best way to hydrate and retain the fluid in the body. Once that was also successful, in the meantime what happened is BARDA, Department of Defense, and Nuclear Countermeasure Program, you know, in case of a nuclear bomb, a lot of people can get radiated, their gut can be damaged, and you will throw up, you cannot take fluids, you cannot keep the fluids in or even the food inside. So there were different formulation that has been shortlisted, and you can see that our formula that we made is also now shortlisted in case of a nuclear event happening in this country. This is actually a work that was actually been done by BARDA and NASA. All this is true. So we said there is an increased stool volume, stool frequency, it's not presenting future episodes. All this is true. Can we demonstrate this in children with severe diarrhea? So again, we went back to ICDDRB, where we first found that stool volume is increasing. So we went to ICDDRB, and we did clinical trial in children. So you can see that these are non-cholera, watery diarrhea in infants and young children. So 6 months to 36 months. And they were sick children in hospital with massive amount of diarrhea. And our idea was this was tested against the WHO Wallace. And do we do better or equal? And this was the first formula ever to remove glucose from the hydration formulation. And we replaced that with the 5 specific amino acid. And this trial was completed, and this now is actually published. You can see that's a phase 3 clinical trial. So that's a story with diarrheal diseases, tools, but any questions in this room? Yes, please.
So what was the result of the clinical trial? How much?
So
better was
in the clinical
solution?
trial, the time to first form stool, the amino acid formulation were faster. Second most important thing, the mucosal mass increased in children. So there is a condition called environmental entropathy. I don't know how many of you will know this. It used to be called tropical entropathy, where children can be stunted, wasted, and malnourished. Not because they don't have food, it's how the food is utilized by the body. And in certain geographical regions and certain regions of the world, children don't perform as well. And the condition is called environmental entropathy. And these children, you know, when you all went to pediatricians, you should ask your mom and dad, they will tell you that your height and weight would have been put across age, a growth chart. And when it goes below 3rd percentile, they say you're malnourished or you're underperforming in terms of your growth. And these children below 3rd percentile were selected for the studies. And we demonstrated that we could improve the height and weight over a period of time. So we did this study in Kenya, we did this in Bangladesh, and now we are in the process of putting another massive study in the same region. So, any other questions? No. So, from this, I'm going to transition a little bit out into peristalsis. Anybody here understand what this term is? Say that again, please. Very good. So, intestinal movement, motility, whatever you want to call this, is peristalsis. I'm going to go into the details. So, I'll ask this question a little differently. Say if you are constipated, and I know you don't want to discuss constipation when you are eating food, but let's discuss that. So, what is the drug or formulation over the counter that you would use if you have constipation today? Any one formula that you can talk about? What is that?
No problem yet.
We don't have the problem yet. Oh, I'm sure everybody has a problem at least once in their lifetime.
Don't get whatever their mom gives them.
So you would have heard about Miralax. You would have seen Metamucil in the store. But you know, none of these things are dealing with pets. I'm going to come into that. So let's go into that. So let's talk about constipation a little bit. What is constipation? Less than 3 bowel movements a week. I don't know, none of us may qualify in that. Stools hard, dry, and lumpy. You want to discuss it here with food? Yes. Stools are difficult or painful and pass, or a feeling that not all the stool has come out. That's also constipation. And I can tell you, this is a progressing thing. How common is constipation? All ages in the United States. 16 out of 100 people, so 1 out of 10 or 2 out of 10 will have this problem. But look at what happens when you turn 60. 33% of the 1/3 of the population is constipated, and we can look at the people and sort of understand who is constipated. So we will build into that too. And that is a gap. So now we have to identify the gap in constipation, correct? So today we have PEG 3350, that is polyethylene glycose, that's Miralax. It's a brand name, by the way. I'm not paid to make them look bad, or I'm not trying to sell anything here. So I'm just putting PEG 3350. It's been there. It has been used, I think, from 1986. Nothing has changed. And how does Miralax work? Can anyone in this room say how does Miralax work? It's a polyethylene glycol. The answer is there in the name. Polyethylene glycol is not absorbed by the body. Anything that is not absorbed, what happens? Will act as an osmotic agent. So it will draw water towards it. So inside the lumen, now it is bulking up. Now there is a pressure effect, everything will come out. But what it is not doing is, it is not addressing the motility of the blood. It's a very passive event. You're putting something into the body that is not absorbed. Because it is not absorbed, a lot of fluid is coming into the body, and now the contents is pushed out by pressure. But did you study peristalsis? Did you study the gut motility? The answer is no. Fiber intake, how does the fibers work? Miralax, no, Metamucil is a psyllium husk. Same thing here, not absorbed, is hygroscopic. It imbibes a lot of water, bulks it up, pressure effect, comes out. Secretory agents, linaclotide, and there are so many other drugs. They secrete a lot of chloride, water into the body, bulk it up, push it out. Side effects, electrolyte imbalance. You cannot use it on a routine basis. So there are limitations. Even PEG has got limitations. Even there are so many abnormalities which I'm not going to go into details. But that's a limitation here. That's a gap. Stool softeners, people use it. A lot of people would have used it. Laxatives, enemas. None of these are addressing the motility. Because how do you study how does the gut move? You know why that has not been studied? Or why a formula has not been developed even in 2025? Because you don't have a tool to study that. So the primary physiological effect is gastrointestinal motility or peristalsis. Okay? So you need to you need to make a device to study peristalsis. It needs to be quantitative. So with this, why is this complicated, right? So this is a cross-section of an intestinal segment. You can see that there is an outer muscular layer, which is longitudinal. Then there are rings of circular muscle fibers, which we call the inner muscular layer. Then you have got submucosa. Now you can also see there are pacemakers, like just like you have in your heart, you have pacemaker. You have multiple pacemakers in the gut, and we call this interstitial cells of Cahal, ICC. Now these pacemakers will sense. So we have this myenteric plexus and mesonous plexus. So submucosal plexus senses the nutrients from the gut lumen, send the signals into the myenteric plexus, then into the central nervous system. That's how it works. Now you can see there is a muscle that moves like this, there's a muscle that moves like this, two different movements. And we don't know who is in the driver's seat. The pacemakers are there, the nutrient sensors are there, but which muscles works first, and who drives this whole movement? So we thought we will make a device, and we put a structure here. The heart of this machine was just a camera, and we had to get a high resolution camera with which would capture those movements, subtle movements. And we put this intestinal segment in an organ bag, and we wanted to capture the longitudinal movement and the circular muscle contractions, and that was captured into a processor that is called the KNCV X400. Then when the muscles contract, the pressure inside the lumen increases. So we had pressure transducers that measured the intraluminal pressure. Now if the pressure goes up, at the very end, the fluid will come out. So we had a detector there that detects how much fluid is coming out, and we had to find out how many microliters of fluid is there in every drop. So all these had to be with a very high level of precision. So I want to show you that machine. We created it. It looks very elaborate. You can see this is a live intestinal tissue in an organ bag. This is how the fluid is coming out. We have a sensor that detects. You have the intestinal segment here. Now you have say, special cameras there. I'm going to show you how these cameras really work. So what does this really have? You can see that it's an horizontal batch system here. It is water jacketed so that it is connected to a water bath that will keep the tissue at 37 degrees Celsius. It is bubbled with 95% oxygen and 5% carbon dioxide so that it is behaving just like it is kept in the human body. Then the outflow, it has to mimic the flow rate that is happening in the body. So that's what we have done with this machine. You can see this water jacketed. The machines looks very complex, but I'm going to show you some pictures which you may like. I'm going to play a video of this intestinal feeding here. You can see how it is bubbled with oxygen, and the curd is now behaving nicely as it's contracting properly back and forth. And then you can also see the blood vessels. And these blood vessels we are going to track. Now, using some digital trackers. So we put some digital trackers. These are virtual trackers built into that camera that is tracking as the intestine is going back and forth. We also put horizontal trackers. We put trackers here and here. As it contracts, we will show how much diameter is actually decreasing. This is what we did. The segment that I showed before is a small gut. Now we have to see the behavior. How does it do it in a large gut? That is the large intestine. If you watch carefully here, you can see even the hostile contraction. Can you see the like sacral formation? Can you see it here? So this is typically how your colonic contraction takes place. And once it contracts, the fluid will come out.
I have a question.
Yes, please.
So how does the flow comes out without the valve?
Say that again, please.
How flow moves forward without having valves in the system?
You said valves?
Valves.
Okay, so
the lymphatics would have valves, and this allows flow to move forward.
Correct, correct.
So here you have a system without valves.
There is no valve. So that is a great, great question. So you can understand that when you eat food, there's a churning action in the body. It has to be mixed. So there are anterograde, which is progressive movements. There are retrograde movements, which is going backwards. Then there are segmental contraction that is just standing there and contracting. Now, this device quantitates anterograde, retrograde, and segmental contractions. Now, the question is, if we have more retrograde movement, the food is not going to go forward. It's going to be constipation or you're going to throw up. If you've got more anterograde movement, everything is going to come out. You're going to poop in your pants. So this is how it is going to work, and we are going to quantitate that, and that's how this machine is going to be built. So that's a great question. There is no valves in the body except there are few of them at the esophageal and gastric junction, then ileocecal valve. But that is separating the small intestine from the large intestine, right? But in the rest of the regions, you don't have any valves. Okay, I'm going to show you another image how we are going to use this light. You can see that we are flickering this light to 50 milliseconds. We are taking a picture, and we are going to digitalize that. And this is how this setup is going to work. Now I'm going to show you how this the movements that you saw before. You can see now it is in a wave format. It is coming as waves. It was not as clean, so we had to get in engineers. And luckily, I was introduced to a friend. You know, this is long before cell phones really came to this planet. You know, this guy, what is it called? Motorola. He worked for Motorola, and he had a couple of patents in Motorola because you know, each wave or what is it called? The cell phones work in a bandwidth, and that bandwidth have a lot of noises. So just the way you are using phones today, the phones were not very clear. So you had to filter away all the unnecessary noises so that the voice that you hear is crystal clear. Similarly, we were getting a lot of junk with the signals that we were capturing. It had to be filtered and cleaned so that we will just capture the oscillation that is coming from the gut movement. And you can see these wave patterns. So this is the intraluminal pressure change. As I said, as the muscles contract, the pressure inside the lumen changes, and you can see it's not steady. Sometimes there are spikes, sometimes it's very quiet. We magnified a small segment, and that is what you see here. And there are definite patterns within the small region, and this is just a magnified view of this. Now you can see there's a crest, there's a trough, and that is the amplitude. From amplitude by time, we knew this is a strength, so we could calculate the frequency of these contractions. We could calculate the amplitude of this contraction. We used a fast Fourier transform. Guess what? And I'm not going to go into details, but if someone is really interested, I can come into details with that. Iron deficiency anemia. You know, a lot of people are anemic, especially women of reproductive age group, people in certain geographical regions, geriatric populations. You can see we put some mice that is deficient in iron. You can see the frequency is faster. They go much faster. And what we found is they are so fast, they're like atrial fibrillation. They fibrillate, they don't contract, and the contraction is so weak, they don't transform into a movement so that you get constipated. So that is a beautiful example I have shown you here. So you can see the blue is iron deficient. The iron deplete are normal iron content in the body. It's a normal diet. So these are the various parameters which we captured using a machine: volume changes, intraluminal pressures, longitudinal contractions, and we had 4 trackers at different spaces, and we could calculate the diameter changes. Putting all the, so coming back to your question, so this is how we found out. So you can see the pressure change. This is the longitudinal muscle contraction. You can see the longitudinal muscle contraction, the activity precedes the pressure change. Can you see that? So this is the longitudinal muscle contraction. You can see that started firing sooner than the change in the intraluminal pressure. So now we understood the longitudinal muscle should move first before the subclavian muscles can contract. Now that was a beautiful finding. I'm going to show you this. So how you should look at this, let me see if I can draw this here. So you have got rings of circular muscles, and then you have longitudinal muscles like this, correct? So the longitudinal muscles will move, and they will gather the rings of circular muscle fibers to one end, and the pacemakers will come together and they will fire and they contract, and that's what we found. So who is the driver's seat? It is the longitudinal muscle. Now, how do you regulate that? That's how you make formulas now. So I'm going to go into that now. So we found out there are changes. Now, anterograde, retrograde, and segmental contraction. This is a typical segmental contraction. You can see that in one waveform and an adjacent waveforms are quite different. At every given time, you can have anterograde, retrograde, or segmental contractions. And you can see there's a net retrograde, net anterograde, segmental contraction. Segmental, all the waves are happening, both proximal and the distal segments are both contracting at the same time. So it's a segmental contraction. So this is now published.
So I have a question.
Yes, please.
So what controls the timing of the action potential at different parts of the intestine? Because the heart has a single pacemaker. It starts and then everything goes to syncytium. But here, in different parts of the heart, or in different parts of the intestine, you have this action potential being initiated at different times.
Good. Can anyone in this room answer this question? And I'll come to that. This is a very, very, very good question, because this is where the physicists should come in. I'm sure you would have studied nodes, antinodes, and things like that. Anyone in this room know that? No. Don't worry. So one of the important things that we found is going from proximal to the distal segment, the frequency of the firings are different. That is the key factor here. So the firing that takes place in the proximal segments are a much, much faster rate. By the time you come to like jejunum and ileum, it's slower. By the time it reaches the colonic segment, it takes minutes. That's the difference. And
That's different segments.
Exactly.
So what segment did you?
we went from all the way from the top to the bottom. You saw that I put
Alright, so
jejunum, ileum, and the colonic segment.
So you did your experiments with different.
Exactly. So we had a series of chambers, and each segment was mounted separately, and each one of them were connected to this tragus, and we studied their patterns. So you can see that the longitudinal muscles are moving in this direction. The rings of circular muscle fibers are contracting. It is gathering the circular muscle, and it goes in for a contraction. So you can see that the distal muscle is contracting. Now the lumen fills up. Now you can measure the diameter change, because we have a camera that detects the volume change. Now, if the proximal muscles are contracting, the longitudinal muscle moves to the proximal end, it gathers the rings at the proximal end. So a proximal contraction and a distal relaxation, foot goes forward. That's an anterograde movement. So this was published in 2023. Now, we use a tool. Can we use as simple things as nutrients in our body, like amino acids, peptides, carbohydrates, but here we have some amino acids. You can see we rank them to see which one of them will bring the fluid out faster. And you can see some of them do. So we rank all the amino acids. We put a formula together, and we want to validate if this is true. So we need an animal model of constipation. So we used how many of, I don't want to use that, it's too sensitive question. So loperamide. When you have diarrhea, use loperamide, and it stops diarrhea sort of immediately. What it does, it constructs a sphincter so that nothing comes out. The toxins stay in the body. So this is loperamide. This is normal, this is loperamide. Now, we discussed Miralax. So we tested this against Miralax. And you can see that our formulation, it just contains food, just amino acid, was performing equal or a little better than the Miralax. So the key factor here we are saying is Miralax, which works by bringing more fluid into the gut lumen and increasing the luminal content and pushing things out. We didn't do that. We just regulated the GI motility. And we wanted to see if we will still do better with without bulking the GI tract. Here we are. So you can see these are very simple, very gentle, and not many complications. And before I go into this, any questions in peristalsis?
So in your lab, you're planning to maybe test trials?
Yeah, so we are getting into
because
yes, we are getting into clinical trials now with this formulation. We are in advanced stages of negotiation for commercializing this particular formula. So, but you know, putting your thinking hat on as engineers, looking at the machine which is very crude and bulky and laborious, you have any simple solutions for us? That is where the brain comes in. So think about it. Now, lung diseases. So now, this is another area. I'm not a pulmonary guy. I'm a GI person trained in gastroenterology. But I thought some of the formulations and some of the channels that I study is the same in lung too. And one of the channel that I'm going to talk about is CFTR. CFTR is nothing but a chloride channel. So in the lung, if you don't secrete chloride, the fluid secretion will not happen, and you have to have fluid secretion in the lung to have the airway surface liquid. And the airway surface liquid is the one that acts as both as a barrier and also for bringing the dust particles out of your body, and even the mucus that you see. So now we went after one of the toughest of the disease conditions called cystic fibrosis, a very, very rare condition where CFTR protein is having some kind of defect. Different types of there, class 1, class 2, class 3, all this. I'm not going into any of the details, but class 1, to this date, there is not a single drug that actually will fix a condition. Oh, you can't hear?
No, from the other side.
Yeah, okay. So in class 1, the class 2, 3, 4, there are formulations that is working. We have a drug called Trikafta that is there. I don't know if anyone knows anybody with cystic fibrosis. A year of treatment with cystic fibrosis with Trikafta will cost approximately $350,000. The drugs are very, very expensive. A disease that used to be a childhood disease now has become, what is it called as, children has gone up to 24, 25, 26. So they're born into adults with the new drugs. They're expensive, but it's working, but that formula does not work in class 1 mutation. Similarly, these drugs have severe complications. They produce liver damage, multiple organ failures with time, and still they get the disease manifestations is still there. So these are the different mutations that I was just talking about. I'm not going to go into the details. Each one, the type of mutations are different. But in the class 1, which is so important because it's a nonsense mutation. There's a stop codon that it prevents the protein from being formed. And there is no protein formed there. So now that's the one that we're going to target. Now, when chloride secretion is not taking place, what happens to the cell? Now, if the chloride ions don't come out through a CFTR channel, more of the chloride ions will stay inside the cell. The cell gets hyperpolarized. It becomes more hyperpolarized, and it is trying to exit, but there is no channel there to exit. But there are alternate chloride channels. So that's what we are going to harness. At the same time, there is an ENAC, which is a sodium channel. They try to bring sodium into the cell. Now, when we wanted to do this, we wanted to prevent sodium from bringing sodium into the cell and stimulate chloride through an alternate channel. That's all we try to do. So Damiano, because you're here, very good. This is your slide.
I didn't make.
So you want to talk about a little bit about this? He will do a much better on this topic than me.
Well, okay.
Yeah.
Maybe 2 minutes.
Yeah.
So when I was recruited by Sagar, I went to there to do what you mentioned, from instead going to bed, back to the bench. Because the formulation they work. Like you imagine also there are many things that work because the Indian has the product from the plant extract. It works, but you never know why. So you need to go back So my work was to record a single cell level, what happened to the currents. In this case, you were talking about the CFTR. So, and we test the formulation. If you want to see if the protein in your study goes to the membrane, you can use a blocker. So when you use a blocker and you see a block of this current, it means that the protein producing the current is dead. So in this patient, because we use in this case, as I mentioned, patient cells, that the stem cells from patient that have the disease. So if you use a blocker for this current, you see nothing because the protein is not there. So I was in charge to use the test the drugs, and when I test the drugs, that formulation, sorry, called drugs, but I don't, it's not a drug. Amino acid, they cost $5, not 350,000. So it was amazing for me. So you use the formulation, and then you use the blockers, and you see the current exists again. That means that the mutation that keep the protein inside the cells went to the membrane. So that was my job in this case.
But can you just describe to them this particular one?
Yeah, but that's the technique is that this is the cell, this is my pipette, patch pipette. You go in the cell, you break in, you can measure the currents going through the membrane because they have amplifiers, they can measure picoamps of current. And when you do this, you can see the increase or decrease of the current. And when you use a blocker, you can see the blocker is specific for the channel you study. And then if it blocks, it means the channel is there. If it doesn't block, it means there is no current. And when you use the stem cells from patient, they have this mutation, and then you see the increase of the current, like the TCAF. I was competing. The TCAF does $350,000 with the amino acid formulation, and had the same effect. So the TCAF bring the current up. I'm not going to explain the whole picture because the story
But I
is
just want to
different.
tell this.
I know, but this.
You can understand this and this.
I can give a talk to you next time about just about this.
Yeah, you can see the channels are closed,
Yes.
or the channels open up with the specific amino acid. So that's fundamentally what it was.
Okay, so
thank you, Damien.
the formulation brings back the CFTR that is mutated.
So the point here is, just like what we did for diarrheal diseases, we ranked all the 20 amino acids for the activity of chloride channel. What amino acid can bring the chloride out of the body? And what will inhibit the sodium, that is the biometric sensitive current, which is the sodium current, which will decrease or increase the sodium current. And based on that, we put a formulation that contains 4 amino acids that actually did the work. This work was published 2025. I'm sorry, this is published in 25. This is again in 24, again on the ENAC function. And you can see how the channels open and close. You can see the reversal potentials and the rectification. So every ion in the body will have a rectification potential, right? That's an electrical signature.
Can I have some?
Yeah, please.
I don't know figure out what's the problem here now. Because this formulation you cannot eat. You have to go in the lung. So the next step, the next challenge is to find a way to deliver the formulation to the lung.
Correct.
And that's why.
So I'm going to show you some pictures here. So again, you can see how you use this formulation to increase the current, and you're using the inhibitor to bring the current down, and that actually proved that we are opening a channel and then we are able to close this channel. And here is the change in, so if you are able to bring chloride out, then there should be water coming out. So then the airway surface liquid volume should be high. So you can see the volume is high. Again, if the fluid is coming out, now there are cilia's inside it, it should be able to flap correctly. So you can see it is flapping correctly. So that movement has increased. I'm going to show you a picture here. You should be able to clear the fluids and the particles from the body. Can you see this? These are particle movements. And now if you put the amino acid, you can see the particles actually move much better. So this is what we have done. So this is why I'm here. This is a cross-disciplinary synergy. There's a clinical relevance, device design and biosensors, the data interpretation, data acquisition. So I believe that communication between the scientists, engineers, and doctors should take place. Then comes the academy and the industry partnership. I think that should also take place, because making a formula is good, but if you cannot take it to the bedside and to the patients and of course commercialize it, then that doesn't make much sense. So you should have a partnership with the engineers, you should have a partnership with the industries. So any questions for this? With that, I'm going to end the show. Any questions?
Does your formulation compete with Gatorade now?
So that, yeah.
Do you have any issues?
We do. So, Gatorade gives $25 million to the campus. On this stage, I'm probably giving $1.5 million to the campus. So they're climbing up the rank. We're doing more studies. So we are just 3 years into the market. So we'll see how it happens. Yes.
We're trying to figure out what amino acids to use in the formulations. Do it just like cell culture or do it with animal models?
So which model are you talking about?
The hydration one.
Oh, fantastic. So remember I drew a crypt in the villi? Good question. In the crypt, you have a stem cell. Now, the stem cells divide, they produce daughter cells. Now, these daughter cells will progressively go up into the villi. And eventually they slough off. So your entire GI tract is renewed by a new GI tract every 5 to 7 days. Did you know this? Every 5 to 7 days, you have a new GI tract. People didn't know this. That is why when you give radiation, what does radiation chemotherapy do? They target rapidly dividing cells. And you have rapidly dividing cells in the crypt. So you destroy them, then you are not going to have a villi. If you don't have a villi, you cannot absorb nutrients. So what is the first thing you will do? The first thing you will do is you will make the stem cell survive. We found the amino acid valine will make it survive. Okay, now if the stem cells are surviving and they are producing daughter cells, what is the next thing that should happen? It should mature and differentiate and should have all the functions a normal cell will do. So mature and differentiation, just like everybody is maturing and differentiating in this room, because that's the progression of life. So we found serine is an amino acid that will help you in maturation. Now, if the cells are maturing and differentiating, what is the next most important thing that you hear about in the internet and all the social media? That is about the leaky gut. Leaky gut is a layman's term. It just means the paracellular space is loose. Once the space is loosened, the luminal contents, bacteria, toxins, partially digested food can go through the space into the blood and produce inflammation. That is fully correct. But how do you fix it? Threonine and tyrosine. These are two amino acids that will tighten the bag. So you've got all your amino acids. Thank you. Any other questions? Thank you.
Thank you, everyone.
Or to Tiaki? Whatever you guys want.
No, it's comments. I stretched it.